네, 안녕하세요. 네, 저는 프로바이오에서 비즈니스 디벨롭먼트를 담당하고 있는 홍진수라고 합니다. 저희 프로바이오가 주최하는 2025년 바이오로직스 심포지움의 진행을 맡게 돼서 굉장히 영광입니다. 오늘 참석해주신 모든 귀빈 여러분들께 진심으로 감사 인사 먼저 드리고 본격적인 심포지움 진행에 앞서 오늘 행사에 대해서 간단히 안내 말씀 먼저 드리겠습니다. 오늘 심포지움은 오후 5시 5분까지 진행이 됩니다. 섹션은 영어로 진행이 될 예정이고 각 섹션이 끝난 이후 질의응답 시간이 주어집니다. 질문 있으신 분들은 자리에서 손을 들어주시면 저희 직원분들이 마이크를 전달 드리도록 하겠습니다. 질문은 영어로 해주시면 부담스럽죠. 저도 이제 영어는 부담스러워가지고 영어나 한국어 중 본인이 편하신 언어로 질문해 주시면은 저희가 조율해 드릴 예정이며 한정된 시간 안에 답변 드리지 못하는 경우에는 저희 심포지움 이후 개별적으로 답변 드릴 수 있도록 하겠습니다. 심포지움 이후에 혹시라도 별도로 미팅을 원하시는 분들이 계시면은 주변에 계신 저희 스태프분들께 전달을 좀 해주시면 저희가 어레인지 후 원하는 주제에 맞춰 미팅 조율을 해드리고 있습니다. 심포지움이 끝난 후에 퇴장하실 때 등록 데스크에서 저희 설문조사 완료 화면을 보여주시면 소정의 기념품을 제공드리고 있으니 기념품도 잊지 말고 받아가시고요. 마지막 섹션 끝난 후에는 럭키드로우 이벤트가 준비되어 있습니다. 소소하지만 굉장히 좀 유용한 경품이 준비되어 있으니까 심포지움 끝까지 많은 관심과 참여 부탁드립니다. 마지막까지 남아 계신 분을 한정으로 추첨을 하기 때문에 혹시라도 자리를 비우시면은 옆에 계신 분이 당첨될 확률을 높여주시는 거예요. 네. 그러면은 오늘 프로바이오 한국 부문장을 맡고 계신 제니퍼 주문경 상무님의 인사 말씀으로 오늘 심포지움을 시작하겠습니다. 큰 박수 한번 부탁드리겠습니다.
네, 안녕하세요. 반갑습니다. 먼저 이렇게 바쁘신 와중에 저희 프로바이오 바이올로직스 심포지움에 참석하신 모든 분들을 환영합니다. 네, 높네요. 빼도 되나? 네, 먼저 최근에 이런 항체의약품 시장이 빠르게 성장함으로써 최근에는 이제 새롭고 다양한 이런 혁신적인 기술을 사용을 해서 새로운 패러다임 시대를 열어가고 있습니다. 기존의 항체 의약품들을 보시면은 면역 항암이나 혹은 이런 자가 면역 질환에서 상당한 많은 두각을 이루었는데요. 최근에는 많은 질환으로서 확장이 되었고 그 중에서도 저희가 신경, 퇴행성 질환이라던가 혹은 감염성 질환 이런 질환으로서 현재 확장이 됨으로써 항체 치료제를 현재 많이 개발을 하고 있습니다. 그리고 이전에는 당연히 저희가 단일 항체 위주로 개발이 됐다면 최근에는 다중 항체, 즉 이중 항체 혹은 삼중 항체로서 개발을 하고 계시고 또 최근에는 트렌드를 보면은 T셀 인게이저가 상당히 핫하게 글로벌리 트렌드로 이어지고 있습니다. 오늘 저희가 다룰 주제 중에 하나인 ADC 아마 많은 분들이 개발을 하고 계시고 또 오늘 관심 있게도 들으실 분야라고 생각을 합니다. 네이키드 안티바디에 저희가 강력한 페이로드를 컨주게이션해서 저희가 지금 캔서 셀에 드러그를 딜리버리를 하고 있는 그런 유발탄을 유도하게 되는 방식입니다. 국내에 많은 분들이 현재 ADC에 관련된 파이프라인을 개발을 하고 있습니다. 오늘 프로바이오는 이런 글로벌 트렌드에 맞춰서 다양한 세션들을 좀 준비를 해보았습니다. ADC 디스커버리 파트와 또 다양한 항체들에 대한 저희들의 경험, 또 이런 나노 파티클, 나노 바디, 그 다음에 저희가 컴플렉스한 몰레큘들, 그 중에서도 바이스피시픽 안티바디에 대한 CMC 경험들에 대해서 공유를 드릴 예정입니다. 프로바이온 지난 6년간 많은 고객사들과 협업을 하면서 또 많은 프로젝트를 성공리에 이끌어 왔습니다. 그래서 오늘 이 심포지엄을 통해서 앞으로 저희가 새로 개발한 바이오 의약품들을 같이 좀 고민해보고 또 중요한 모멘텀이 될 수 있는 하루가 되기를 바라며 네, 심포지엄을 시작하도록 하겠습니다. 네, 감사합니다.
네. 첫 번째 섹션은 프로바이오 이제 차이나 난징 사이트의 안티바디 드럭 디스커버리 부분의 수장이신 데렉 첸 님께서 발표해 주시겠습니다. 원스톱 솔루션 포 ADC 디스커버리라는 주제로 발표해 주실 거고요. 데릭 님에 대해서 간단히 소개해 드리면 면역학 박사 학위를 보유하고 계시고 10년 이상 항체 의약품 개발에 대한 실무 경험을 가지고 계십니다. 이러한 경험을 바탕으로 세계의 바이오 파마슈티컬 특허를 보유하고 계시고 현재까지 200개 이상의 프로젝트를 성공적으로 이끌며 저희 프로바이오의 항체 개발 서비스를 이제 중추적인 역할을 담당하고 계십니다. 큰 박수로 대리님을 환영해 주시기 바랍니다.
All right. First of all, thank you everyone for taking this seminar. As Jinsu mentioned, the first topic of this seminar will be the one-stop solution for the ADC discoveries. Let's begin with a very brief overview of the ADC landscape. So this table summarizes the so far 7 launched ADC products for blood tumor, the hematological malignancies. Most of these drugs are approved before the year of 2020, and they are approved for most of them are approved for the indications including like myelomas, leukemia. So this is for the first part, and the second table summarizes the 8 drugs currently marketed for the solid tumors. And the majority of them are approved after the year of 2019. As you may see from these tables, the solid tumor is gaining more and more interest in ADC markets in the recent years. As you may well know, the ADC is comprised by 3 parts: the naked antibodies, the payload, and the linkers. For the discovery of the naked antibodies, usually we will focus on the profiling of 6 different aspects, including the antigen binding specificities, affinities, the cross-binding to cynomonkey antigens, the internalization efficacies, the biophysical, biochemical properties, which is developabilities, and the immunogenicities. In addition to the naked antibodies, the selection of the payload and linkers has to be made based on your target MOAs, the target biology, the disease biology, including the toxicities, the hydrophobicities, the PK profiles, and etc. In order to give a comprehensive profiling of your ADC products, after the conjugation of the ADC drugs, usually we will evaluate the candidates from different perspectives, including in vitro assays, in vivo disease models, and also the general developabilities. The discovery of ADCs workflows can be categorized into 7 stages. The first stage, the target discovery. Usually this is a time to define your target product profile, the TPP, based on your target biology, the disease biology, and the indications, and also the competitive landscape. Once the TPP is defined, we will move on to the second part, the naked antibody discoveries. People commonly use a variety of platform technologies like hybridomas, phage displays, and a single B cell to generate antibody leads for the ADC candidates. And during this stage, the assessment of internalization is a very crucial step to narrow down your candidates for the ADC applications. At the same time, we'll also start to think about the selection of a payload and linkers. Once both parts, the naked antibody, payload, and linker are decided, we'll move on to the stage 4, the ADC conjugations, using technologies like splicing or cysteine-based semi-random conjugations or the site-specific conjugation technologies to link the payload to the naked antibody carriers. Following the conjugations, we will evaluate the ADC candidate by in vitro assays, in vivo disease models, and also the developability analysis for stability assessment. Okay, next, let's start with the naked antibody discoveries for a more detailed discussions with the workflows we just introduced in the previous slide. So far, the technologies used for naked antibody generations can be categorized as the two major types: the animal immunization-based, or called the in vivo-based approach, and the in vitro-based approach. Both in vivo and the in vitro approach can be achieved by platform technologies like hybridomas, single B cell, and phage displays. And the final leads can come in as the formats like traditional full-length IgGs or antibody fragments, including Fab, single domains, or single-chain FVs. Among these different technologies, hybridoma is the one we recommended for the specific ADC applications, the screening of antibodies for ADCs, because using this hybridoma-based platform, or we call the PowerDoma, you can use the hybridoma supernatant to conduct the high-throughput internalization assays at a very early stage. These particular features can significantly improve your project's success rates and reduce the overall timeline and the cost. So basically, it can help you narrow down the antibody binder candidates based on the internalization kinetics, which is a necessary indicators for the ADC candidate. Speaking of the internalization, currently there are 4 different approaches to quantify or to measure the internalizations, which is the live cell imaging systems, the toxin conjugated approach, the pH indicator, the pH rodel, and the temperature shift-based assays. The top 2 ones comes with the high throughput and automatic platforms. So these tools are the ones we usually recommend it for discovery stage for the purpose of early stage high throughput screening, because it can run through hundreds, even thousands of samples and provide much better signal sensitivities and assay robustness. Let's go over these 2 different assays one by one. First with the live cell imaging systems. So this assay is achieved by a hardware called InQSight. I believe some of you guys are already familiar with it. So the net rationale of this hardware is to utilize a pH-sensitive fluorescence dye. And when the antibody take this dye into the cell, into the lysosome, the acidic conditions in the lysosome will trigger the fluorescence dye and make it start to glow. You can start to, and the InQSight will start to collect the fluorescence signals. The intensity of the fluorescence signals is proportional to the rate of internalizations. So basically, this is how we determine the internalization based on the fluorescence signal intensities. The key feature of this platform is able to evaluate, to measure the internalization at 2 different levels. Traditionally, people do this kind of assays by a dose response curves, like the graph to the bottom right. So basically, we can establish a correlation between the antibody concentrations on the axis and the internalization intensities on the y-axis. So this is a typical dose response curves. In addition to this, this InQSight live cell imaging system can also provide a time course-based analysis. On the x-axis, it's a scale by hours, and on the y-axis, it's the intensity of internalization. Basically, this analysis can provide the kinetic data of each antibody internalization on a specific cells on a specific time point. Alternatively, we can use another high throughput system. It's called DT3C conjugation systems. So DT3C is a synthetic toxins. It's not very commonly used for ADCs, but it is commonly used as a reagent or as a tool to measure the internalization. Similar to the pH-sensitive dye, this toxin can associate with antibody constant region non-covalently. So basically, you do not need to do any chemical reaction to conjugate the toxin to the antibodies. It's a simple mixture and incubation process. So after the incubation, the antibody will take the toxin into the cell and start to kill the cells. By the measurement of the cell death, we can convert the cell death to the relative internalizations. And the key feature of this platform is it's the high throughput systems. You can do it in either 96 well plates or 384 well plates. It's pretty similar to the traditional Fab-Zap systems. The only difference is it provides much higher throughput. So it's particularly useful when it comes to earlier stage, larger scale binder screenings. Once the naked antibody is determined, the candidate is determined, we'll move on to the ADC pharmacology. Start with the payload linker conjugations. So as I mentioned at the very beginning, the selection of the payload and the linker has to be made based on the MOAs. And the ProBio offers a large libraries, including more than 150 cytotoxins and more than 950 linkers to select from to choose the right combinations for the ADCs. And in terms of the conjugations, ProBio provides the traditional cysteine lysine-based semi-random conjugations and also the site-specific glycan side chain-based conjugations. I will share a case studies to compare these two different conjugation methods with a GPRC5D case studies in the large part. So after the conjugations, the first bioassay, the cell-based assays, is the most traditional cell-killing assays. The cell killing can be measured by either like a LDH or CTGs or CCK8. In addition to cell viability assays, you can also do the cell apoptosis assays or cell cycle assays to evaluate the drug efficacies. In addition to the cell killings, bystander effect is another crucial experiment for some of the ADCs. These can be achieved by the same machines I mentioned at the very beginning for the internalization, the InQ site. Basically, in these assays, the HER2 positive cells are labeled with a red fluorescence, and the HER2 minus cells are labeled with a green fluorescence. Both cells are incubated together along with your ADC candidates. And the InQ site will measure the cell death by the signal intensity of a green or red fluorescence. And the decreasing signal of these fluorescence is correlated with the cell death. And if you see the signal reduce with the green fluorescence, that means the ADC has a significant bystander effect, which can kill the HER2 minus cells after entering the HER2 positive cells and releasing from the HER2 positive cells. So after these two typical cell-based assays, we'll move on to the next part, the in vivo studies. It's a pretty standard oncology the studies, including the efficacy, the tumor model studies, the PK study, including both in ex vivos and in vivo PK studies, and also the toxicity studies. For ADCs, the toxicity studies is pretty crucial because most of the ADCs has these toxicity issues, the risk. So that's why we normally recommend you to conduct the early stage of toxicity studies at the discovery stage using either rodents or a non-human primate. To conduct the PK and ADC studies, you need a panel of reagent antibodies like anti-EDO type antibodies. And the generation of anti-EDO type antibodies could be very time consuming. Most likely, you will probably need 5 to 6 months to generate these reagent antibodies to develop the PK and AD assays. ProBio offers a panel of off-the-shelf, ready-to-use anti-EDO type antibodies specific for the naked antibody part and the small molecule part, so it can be easily paired with each other for the purpose of PK and ADA studies. So once the initial ADC candidate is characterized and finalized, we will move on back to the naked antibodies for engineering and the final developability assessment. Humanization is a pretty common antibody engineering approach nowadays. It's really quick, only takes about 3 weeks, and usually it comes with the guarantee of affinities. Affinity tuning is an optional process depending on the antibodies, the affinities of your parental antibodies. If you want to increase the affinity and to increase the toxicities, you can do affinity maturations. Alternatively, if you want decrease the toxicities, you can do affinity attenuations to decrease the affinities. The third part, FC engineering, is not a very popular topic in terms of for ADC discoveries because so far most of the approved ADC drugs use the wild-type ADCs. However, some people may prefer using the FC enhanced FC effector mutations to add ADCC or CDC cytotoxicity to the ADC toxin-based cytotoxicities. Alternatively, some people believe that the FC-mediated macrophage internalization could be a possible reason for non-specific toxicity of ADCs. So in that circumstance, people may want to mutate the FC regions to silence the FC effector function to avoid the FC-mediated internalization, like the phagocytosis. Once the antibody sequence is finalized, we'll move on to the developability assessment. Both the naked antibody and the conjugated ADCs needs to be assessed for its stabilities under different kinds of stressful conditions, including like high temperatures, repeated freeze-thaw cycles, and acidic conditions. Okay, so for the last part, let's have a review for case studies. So this case study is a discovery of ADCs against GPRC5Ds. GPRC5Ds is a very popular tumor-associated antigens in recent years in both cell therapies, in T-cell engagers, and also in ADCs. It is a typical 7-transmembrane GPCR proteins, which is considered as a really challenging target because most of the time you cannot produce high-quality recombinant proteins for the animal immunization to generate antibodies. So that's why people often rely on DNA immunization or whole cell immunization to generate antibodies against the GPCR targets. However, DNA and cell immunization is relatively low efficient in terms of the generating antibody binders to these multiple transmembrane targets. So as a result, in these case studies, we use mRNA instead of DNA and whole cell to immunize the mice for the antibody generations. So after the hybridoma fusions, we screen the hybridomas with both engineered overexpressed human and cyno GPRC5D cell lines, and also the myeloma MM.1S cancer cell lines for confirmation of actual cell surface bindings. In addition to the antigen bindings, we also screen through thousands of binders with the internalizations, the one I mentioned previously, using either the IncuCyte or the DT3C systems to narrow down the candidate to roughly 50 hits. And these hits were subcloned and confirmed by binding and internalization. After sequencing, we narrowed these candidates down to 21 unique leads. And based on the sequence analysis, we selected 7 of them for further characterization and the payload conjugation and also the ADC pharmacology characterizations. Next, I'm going to share a series of detailed data for these case studies. First of all, it's the efficiency of the mRNA immunizations. So the purpose of this study is to compare the efficiency between the mRNA versus the DNA plus whole cell immunization. As I mentioned previously, the DNA and whole cell immunization is considered relatively lower efficient in terms of generating antibody binders. So in this experiment, we compare these 2 different kind of antigens at 3 different levels. The first is by in vitro transient transfection of mRNA versus the DNA. The light green is for the mRNA, the dark green is for the DNAs. And we use 3 different reference antibodies to quantify the cell surface expression level of a GPRC5D transmembrane proteins. As you can see from this data, the mRNA can express the GPRC5D at a much higher level on the cell surface comparing to the DNA in the in vitro transient systems. And the second step, we compare these 2 different kinds of antigens during the immunization stage by comparing the serum, the animal serum titer data. The calculation of the titer is determined by the ratio of flow cytometry intensities, the MFI values, on the antigen positive cell line versus the antigen negative cell lines. So that's why it is a ratio. The mRNA immunization after 3 rounds of mRNA immunization, it will create a cell titer ratio at more than 600, while the DNA plus cell immunization can only generate a cell titer ratio at about 4-fold. So there is like at least 150-fold differences between these 2 different kind of antigens. So this further demonstrate that the capabilities of mRNA to induce the immune responses to generate much more antibodies comparing to the DNA and the cell approach. The third level on the right panel is basically by counting the total number of positive antibodies after hybridoma screenings within different affinity range. The affinity range is calculated with a similar approach as the serum titration data. Basically, we compare the flow cytometry MFI value on the antigen positive cell line versus the antigen negative cell lines to calculate the ratio. And we have a different range of a ratio here, 1 to 5, 5 to 10, 10 to 20, 20 to 50, and above 50s. And within each range, we counted the total number of positive binders. The top 2 rows are the animals immunized with mRNA, and the bottom two rows are the animals immunized with DNA and cells. So based on the number here, you can see the mRNA is able to generate a lot more, hundreds of more positive binders comparing to the DNA and the plus cell immunizations. So after the screening process, as I mentioned in the previous slides, we selected the final top 7 candidates. And first, we evaluated the affinities, the cell binding affinities using overexpressed engineered cell, the myeloma cancer cell, the MM.1S cell line, and the transient expressed cell with the expression of a Cynos GPRC5Ds. In all of these 3 experiments, we see comparable or better cell binding affinities from these 7 candidates, comparing to the clinical benchmark. In addition to the antigen binding affinities, we also evaluate the internalizations with these naked antibodies using both InQSite at 24 hours and the DT3Cs at 72 hours. So these assays are done against the GPRC5D overexpression engineered cell lines. As you can see from these 2 figures, all of these 7 candidates shows similar, equivalent, or better internalizations comparing to the clinical benchmark, both of these 2 clinical benchmark in these 2 independent systems. In terms of internalization, in addition to the engineered cell lines, we also did it with the myeloma cancer cell lines, the MM.1S. So since the GPRC5D antigen densities on these cancer cell lines is significantly lower than the overexpression engineered cell line in previous slides, so the overall signal you are seeing in this page is relatively lower than previous page. But however, we're still able to see a robust internalization of these 7 candidates, which is equivalent to the clinical benchmark. So after careful characterization of naked antibodies, next we'll move on to the evaluation of the ADC pharmacologies after the conjugation with 2 different approach. Here, the G stands for the glycan side chain based site-specific conjugation, while the P stand for the cysteine-based semi-random conjugations. So both sets of samples, we analyze them with in vitro assays and in vivo disease model, the tumor models. So there's a lot of data in these figures. It's just too crowded to differentiate from one sample to another. So we separate them into 6 different group. In each group, it's the same ADC candidate conjugated with 2 different conjugation approach. The P stand for the cysteine cysteine-based semi-random conjugation, and the G stand for the glycan side chain-based site-specific conjugations. And first, we evaluate the distribution of species of the ADCs in different DAR range from 0 to 8, to DAR 0 to DAR 8. As you can see from the top left tables, with the cysteine-based conjugation approach, you see a various distribution of the ADC product. The DAR4 takes about 43% of the total species, but there are significant amount of ADCs distributed on the DAR 0, DAR 2, DAR 6, and the DAR 8. The average DAR is about 3.4. However, when we switch to the glycan-based site-specific conjugations, the DAR4 species takes about 83 to 84%, and the remaining are the DAR2 species. So this data suggests a much better homogeneity with the site-specific conjugations comparing to the traditional cysteine system-based semi-random conjugations. Next, we take these two samples for further pharmacology assessment, including cell-based assays in the middle panels and animal-based tumor model analysis on the right panels. So surprisingly, in the cell-based assays, the system-based conjugation approach gives a better killing efficacies comparing to the glycan-based site-specific conjugations, as you see in this middle graph, the green curve versus the blue curves. This is a little bit of a surprise to us, but in the disease models, in the tumor models, we see the reverse patterns. The site-specific conjugated samples shows a much better tumor inhibition, which is red, comparing to the blue one, which is the semi-random, the system-based conjugated samples. And we'll move on to the next samples, the clone number 53. The same two different conjugation approach, the same distribution analysis for the DAR distributions. We see similar patterns. For the site-specific conjugations, almost 80% of them are DAR4s, but for the system-based conjugation approach, only 43 of them are DAR4s. And again, in the cell-based assays, we see these two drugs showed similar efficacies. However, the semi-random conjugated samples shows a little bit better Emax, but a little bit higher EC50s. But when we switch to the tumor models, the in vivo studies, again, we see a similar patterns like the first candidates. The site-specific conjugated samples, the brown one shows much better tumor growth inhibitions comparing to the system-based conjugated samples. The similar profilings, we also seen similar profilings with another several candidates, the clone 52 and the clone 402 and the clone 390s. Similar or worse in vitro killing from the site-specific conjugated samples, but much better tumor inhibitions in vivo models. But out of these 6 samples, the clone 411 showed a reverse patterns. The cell-based killing efficacy is pretty much similar, but this time the site-specific conjugated samples showed a worse tumor growth inhibitions comparing to the system-based semi-random conjugated samples. So based on the data, let's first look at the cell-based killing assays. Although the site-specific conjugations can generate samples with much better homogeneities, but our theory or our guess is those high DAR species generated from the random conjugation methods has a better potencies in terms of the cell killings. That can explain why we see better cell killings with those system-based conjugated samples. However, in the in vivo animal models, 5 out of the 6 samples showed better tumor growth inhibitions with the site-specific conjugations. So we believe that the homogeneity of the samples may have a bigger roles in actual tumor tissues. In addition to the homogeneity, the antibody itself may have certain functions in terms of these discrepancies of in vivo tumor growth between the site-specific conjugations versus the system-based conjugations. Okay, so that's pretty much everything for my talk. Let's move on to the Q&A session. Any questions?
네, 혹시 질문 있으시면은 시간 관계상 먼저 한 개만 받도록 하겠습니다. 네, 따로 질문 없으시면은 첫 번째 섹션은 여기서 종료하도록 하겠고요. 혹시라도 질문 있으시면은 저희 이제 브레이킹 타임을 좀 이용해 주시면 감사하겠습니다. Thank you, Derek.
Okay.
네, 두 번째 섹션이 바로 이어서 시작이 될 거고요. 두 번째 섹션은 Challenges and Solutions for Complex Biologics (CMC)라는 주제로 잉리 님께서 발표해 주시겠습니다. 잉님은 중국 난쟁의 안티바디 프로세스 디벨롭먼트 부분을 담당하고 계시며 화학공학 분야의 박사학위를 보유하고 계십니다. 머크의 R&D 매니저로 근무한 경력이 있으시고 10년 이상 CMC 다운스트림 프로세스 개발, 뉴 테크놀로지 플랫폼 개발, 바이럴 벡터 매니팩처링 등의 프로젝트를 진행한 경험을 바탕으로 저희 프로바이오의 공장 개발팀을 이끌고 계십니다. 네, 큰 박수로 잉님을 환영해 주시기 바랍니다.
Hello, good afternoon, everyone. It's my pleasure to join today's symposium. And today my talk is around the complex biologics CMC. So first of all, I'd like to show a brief introduction of our ProBio CMC platform. So over the past 9-10 years, we have accumulated a number of CMC case study in terms of track record. As you can see, the different modality distribution across the monoclonal antibodies and bispecific, trispecific antibodies, recombinant proteins, and fusion proteins. So one of the highlights of our platform is relatively comprehensive experience with complex modalities, such as about 30% of our track record belongs to bio-trispecific antibodies, with around 20% belongs to different type of proteins. And over the years, we have successfully delivered over 55 R&D clearances, with most advanced projects has obtained emergency use authorization during COVID period, and two marketed projects, medical devices with ProBio as antibody supplier, and over 200 cell line development projects for R&D applications. Currently, there are two PCPA late phase CMC projects ongoing, with expected first commercialized product by the end of this year. Here, I'd like to show a library of different bispecific antibody formats that we have successfully delivered in the past. So in general, we can see we can categorize the bispecific antibody into the IgG-like, which contains the Fc fragment, and non-IgG-like, which contains only the Fab fragment. So over the past years, we have successfully worked with over 30 different bispecific antibody formats, including such as the IgG-like ones with symmetric bispecific antibodies, which IgG fused with single-chain FV, or IgG fused with single-domain antibody. And for asymmetric bispecific antibody, we have worked with different type of bispecifics with 2, 3, or 4 different chains. For example, as you can see, some of the formats, for example, this trispecific antibody contains 4 different chains with 2 different heavy chains and 2 different light chains. And the second example shows here is FC plus single chain FV with 2 different chains on the A chain and B chain. And similarly, there are a number of different formats as listed here with some examples. And in addition, we have worked with a type of unconventional molecules, such as those does not contain Fc fragments. For example, the single chain FV linked with a Fab fragment, different type of VHH connected in tandem. Some of them has already obtained IND from FDA as an NMPA. In general, the average titer of our platform achieves greater than 6 grams per liter, and average yield is about 50%. And this slide shows a library of different protein molecules. As I mentioned, we covered different type of fusion proteins, such as the Fab fusion proteins with antibody VHH fragment fused with protein or Fab fused with protein. The more common types include different type of Fc fusion proteins. And in terms of recombinant proteins without any tags, we have done CMC projects with the spike proteins for vaccine applications, the hormone molecules, this one is currently in phase 3, and enzyme molecules with instability properties and blood factor 8. This project is currently also in clinical phase 3, and we expect commercialization soon in China this year. And last but not least, I'd like to mention that we have seen an emerging type of applications of antibody fragments, such as a Fab fragment, as single-chain FV fragment, and VHH fragment. And we are going to touch base on this in our second talk this afternoon. There are a number of challenges associated with these different type of challenging molecules. For example, for bispecific antibodies, the most common challenges we have seen include the formation of different byproducts, such as homodimers for asymmetric bispecifics, and a high amount of aggregates and its removal, fragments, how to improve its expression level, as well as the N-glycan optimization. For recombinant proteins, the common challenges include the low protein stability and how to design the capture step for those molecules without Fc fragment, removal of high level of HCPs, the low expression levels, as well as the analytical method development for special protein molecules. Over the years, since we have accumulated a lot of experience in dealing with these challenges, we have established our toolbox platform over the past years. So basically that covers a number of different units, from cell line development, cell culture development, purification, formulation, as well as analytics, and summarize our solutions, platform solutions into this toolbox platform, so that we can tackle those challenges more specifically and more effectively. And over this year, we have also implemented AI function in our toolbox platform, so that we create a ChatGPT version internally to effectively address common CMC challenges with case studies. So today, I'm going to focus on 4 common CMC challenges in those challenging molecules, including how to improve product expression, how to control the byproducts, control aggregates, as well as the N-glycan profile optimization. So let's start with the titer improvement. So basically, how to get a higher titer expression is one of the key goals for the cell line construction as well as cell culture process optimization. So starting with the cell line part, there are a number of key factors that contribute to different level of expression, such as the host cell, the vectors, as well as its selection system. So let's start with the vector selection. So basically, the expression vector is a critical factor influencing the transfection and transcription of our gene of interest. And it will also impact the stability, solubility, as well as the secretion level of our target protein. So in general, the key components of a vector include a promoter, a signal peptide, a different the titer enhancers, or most commonly known as the transposon system, common strategies like a lot of platform has utilized. So here, ProBio has developed over the years a number of different vector systems. So our second generation UP vector, we implement a titer up enhancer element, transposon element, basically that helps to improve the integration efficiency and significantly improve the titer of complex molecules. And over the last year, most recently, we have launched our also proprietary third generation Max vector. On top of the UP vector, we also include a stability enhancer element in our Max vector, where we also significantly enhance the stability of our vector, and allows also for faster recovery in the stable cell pool stage. This application also has filed for IP globally. So here, as you can see, the selection of different vectors could has a huge impact on the expression level of our protein and antibodies. Also, the high throughput selection system also plays a key role in getting high expression single clone. The traditional methods such as limited dilution or FACS suffers from their throughput. So sometimes it's not quite effectively to identify the truly high expression single clone. So over the years, there are a number of technologies to advance different type of instruments to allow for more high throughput screening for high expression clones, such as a number of systems I listed here, the VEPS, the Wolf system, utilizing some systems utilizing the microfluidic sorter, such as a Beacon and single cell printer. So our system also utilizing some of this advanced instrument. Plus, over the last year, our R&D team also developed a fluorescent-based high-throughput screening system. So basically, combined with cell screening utilizing microfluidic cell sorter, we utilize a fluorescent dye that could bind specifically to target protein of interest. And during the screening phase, the system would automatically pick the cells that could express high intensity of the fluorescence dye and more effectively pick up the high expression single clone. As shown in the graph in the middle, compared with more conventional methods such as using VEPS system, our high throughput fluorescence dye-based system could effectively improve the expression level of the selected single clone with a much shorter selection period of time. And moving on to the cell culture stage, there are a number of ways to improve the titer expression. With a common strategies listed here, we can do the media screening and feed and additive optimization, plus the process parameter optimization in the bioreactor stage. And oftentimes, if we are still not satisfied with the titer, we can utilize intense fed batch or the perfusion cell culture mode to further improve the titer. I wouldn't go through details in this because they are very commonly used strategies in upstream optimization. So I would like to highlight our in-house media capabilities. Our R&D team over the last 3 years has come up with our unique in-house media component that suits for our whole cell plus vector system. So basically we start from zero because we don't know the catalog media components that are commercially sold in the market. So starting from scratch, we developed our in-house media platform. And as you can see, we have tested and utilized in a number of CMC projects, over 26 projects. Our in-house media achieved over 70% of the projects achieved the highest expression titer among the major domestic as well as international brand media, such as the Thermo Fisher or Merck, Cytiva branding cell culture media. And the bispecific antibody and mAb titer expression in fed batch mode could be achieved as high as 10 gram per liter and 14 gram per liter. And moreover, I'd like to highlight that over this past few years' experience developing this in-house media platform that give us a very deep know-how with respect to how does different components in the cell culture media impact the cell culture performance, as well as how can we tweak the different components as well as their concentration to get a desired titer expression as well as a quality profile, such as how to adjust different acidic or basic charge variants or different glycosylation patterns. Moving on to the second topic, how to control the homodimer. So the generation of homodimers, such as misparent species, is one of the most challenging topic in asymmetric bispecific antibody. When tackling this problem, we can start with different aspects. So first of all, in terms of the molecular design, actually this is one of the key that could help alleviate this type of challenge. Here, at least a typical bispecific antibody with 4 different chains, 2 different light chains and 2 different heavy chains. Because these chains are freely expressed in the CHO cell, and if you allow a free assembly of different components, it results in a library of different misparents of the species, and the correct assembled bispecific antibody only account for like around 10% of all these different type of molecules. So in general, most of the companies would adopt a type of different designs to prevent the misparents of heavy chains or light chains. So here, at least a number of different commonly used strategies to prevent the misparents, such as knob into hole. I think nowadays the majority of the bispecific antibody adopt this type of format, as well as so the general strategy to prevent the misparent of heavy chains include introducing point mutations in their CH3 regions, either electrostatic repulsion or steric kind of hindrance to prevent the misparent of heavy chains. Some examples include the ART-IG, the dual body with controlled fiber-armor exchange, and asymmetric type of formats. So they all adopt similar strategies to prevent the misparent of heavy chains. And in terms of the misparent of light chains, commonly used strategies include the common light chains or cross map, basically switching the CH1 and VL domain, the light chain domain to prevent the misparent of the light chains. And moving on to the cell line stage, there are a couple of ways to help improve the correctly paired species. For example, our platform commonly adopted a dual vector system to improve both the productivity as well as the purity. Here are a couple of examples of asymmetric bispecific antibodies with different domains expressed in A port and B port. So one of the strategies is to put everything into one single vector. Alternatively, you can also design the molecules expressed in the separate vectors. For example, in this number of cases, we can basically express the component A and component B in 2 different vectors. And by tuning the different vector ratios and further optimize this aspect, we can achieve a higher expression titer as well as reduced homodimer species. Here is one of the example. So for example, this is a 2-chain asymmetric bispecific antibody. So initially, if we put the 2 chains expressed in a single vector, since the B vector is quite overexpressed, even though it's put in a second order. However, we still see the correct pairing of A plus B only accounts for around 20% of the expressed species, and a lot of impurities come from the overexpressed chain B. As a result, the overall titer is also low at less than 1 gram per liter. And with further optimization with the dual vector system and further optimize the vector ratio, as we can see, we can significantly improve the purity level to over 70% and improve the titer by 10-fold. If still we cannot tackle all the challenge of this homodimer species, we move on to the purification stage. Sometimes it's not an overall optimal strategy because oftentimes in order to remove these homodimers, we need to sacrifice some of the downstream yield. Here we give an example of separating the homodimer from our target molecule. So to analyze the purification of different species, we start with analyzing the different chemical and physical properties of the two chains. For example, in this particular case, when we run an IC-IEF analysis of our target proteins, we can see the chain 1 and chain 2 have relatively big difference in terms of their isoelectric point. So delta pi is close to 1. So in general, we think if the two chains delta pi is greater than 0.5, there's a good chance of utilizing electrons charge variance or the CEX or AEX type of chromatography to separate them based on their electrostatic difference. So it is indeed the case. We utilize a mixed mode CEX resin to successfully remove almost all the homodimers in one-step chromatography. And overall yield is 60% because the other 40% loss belongs to the homodimer species. This is another successful example. Again, for this molecule, it designed a knob-into-hole structure with FC fused with two single-chain FV species, asymmetric structure. And in our experience, even though knob-into-hole structure is utilized, sometimes we still see quite significant amount of homodimer expressed, especially with the whole-hole type of mispairing. And as a result, we have seen about 20% of the aggregates because of the introduction of single-chain FV species, as well as around 30% of the whole homodimer that need to be removed. Plus, this molecule is not a stable one. Upon the CEX elution at high salt concentration, the molecule is quite unstable to form additional aggregates. So for this particular project, we utilize a different type of mixed mode chromatography to successfully remove both the high molecular weight as well as the whole homodimer. And for the instability of the molecule, we actually exchange the UFDF and nanofiltration step to perform the buffer exchange first to a stable condition and followed by nanofiltration. So in this case, the CHT resin, the ceramic hydroxyapatite, was used for high molecular weight aggregates removal. And in our experience, the CHT resin, even though it's sometimes conveniently used for certain cases, but it's quite effective in removing some aggregates, especially the ones associated with single-chain FV species. And the mixed mode CEX was used for whole homodimer removal. As shown in this graph, the second step of the CHT resin is very effective in removing all of the high molecular weight in a single step chromatography, and followed by a capital MMC step, which successfully remove all of the homodimer species with a decent overall recovery. Next, let's also talk about how to control the aggregates. I think if anyone has worked with bispecific, we will soon realize that the aggregates control and removal is one of the key challenge in a lot of bispecific antibody projects. So actually, there are a number of different type of aggregates and their underlying mechanism of formation. For example, the aggregates can be categorized into covalent versus non-covalent bond aggregates, or they can be reversible and irreversibly formed, and they can be soluble or insolubly formed. And there are a number of factors that contribute to different level of aggregates in a particular molecule, starting with their the protein structure itself, its primary structure, how many hydrophobicity amino acids are included in their sequence, as well as their secondary, tertiary structures, as well as the post-translational modifications can all contribute to different level of aggregates. In terms of the cell culture, the cell culture pH, DO temperature, the osmolality, different type of media chosen, and redox condition can all impact the level of aggregates. Similarly, in the purification stage, different pH, conductivity, and buffer of choice, freeze and thaw process, as well as shear force, can all contribute to the formation of aggregates. In order to better control the aggregates level, I will also go through 3 different aspects, starting with the molecular design, as well as both the upstream and downstream optimization to minimize this challenge. So first, let's start with the design of the molecule. And a lot of times when we see bispecific antibodies or fusion proteins, sometimes the formation of the design of the molecule itself has a huge impact on the developability. For example, in this particular case, this is IgG antibody fused with C-terminal fused with 2 single-chain FV fragment. And with their wild-type design, we can see it's quickly forms a high level of aggregates when the molecule start to concentrate, which indicates a poor drug developability because we cannot concentrate about 1 milligram per milliliter before we start to see an increased level of aggregates, even without selective pressure. So luckily, this molecule is in the early pre-CMC stage, so we work with the client to help modify the design of the molecule. Specifically, we've seen single-chain FV is very commonly utilized a component to construct a bispecific antibody format. So in general, there are different ways and designs to optimize the single-chain FV design. For example, we can introduce interdomain disulfide bond to help stabilize the structure stability and minimize the exposure of hydrophobic region of the single-chain FV. Alternatively, we can also alternate the design of the linker with different lengths to achieve a balance between the flexibility versus the conformational stress. As you can see, after modification, this molecule is much more stable upon concentration and is able to move next to the CMC stage. In terms of the cell culture, there are a couple of chances that we can successfully reduce the aggregates level. For example, in this case, we first start with analyzing the origin of the aggregate species. For this case, initially, when we first do the first round of cell culture, we see a significant level of aggregates present with over 90%. And upon further analysis, when we run the reduced versus non-reduced gels, as we can see, on the reduced condition, almost all the aggregates disappeared. That's a pretty good indication suggesting that the majority of the aggregates has to do with the covalent bonding. So as a general strategy, we should consider adding some reductants to break down the intermolecular disulfide bond to reduce the level of aggregates formation. So in this project, we did a DOE study of different levels, concentration of cysteine as a reductant and combined with different media and cell culture temperature, and successfully reduced the level of aggregates from over 90% down to 12%. And also the selection of cell culture mode could also influence the aggregates formed in the upstream process. One common strategy is to consider using perfusion cell culture to reduce the level of aggregates, because first, in general, as suggested also in the literature, running perfusion mode could alleviate the overall oxidation of glutathione induced by oxidative stress, thereby reducing the disulfide bond formation and the dimer formation. And additionally, since its perfusion allows for shorter residence time of our molecule in the bioreactor, it oftentimes could reduce the level of non-covalent bonded aggregates as well. So here is one example of shifting different cell culture mode can reduce non-covalent bond aggregates by over 10%. And currently there's one project ongoing, which is quite challenging. We also switch from the intensified fed batch to the perfusion mode, where we reduce the level of aggregates by over 20 to 30%. Again, if we still have additional aggregates to be removed, we are moving further on down to the downstream purification stage. And our platform has established quite a number of effective strategies as a platform solution to tackle the aggregates removal. Here I just want to give a couple of examples. One of this format is a Fab plus single chain FV. This molecule does not quite have a good developability properties. And actually we work with this client with a number of different formats, but similar to this. And none of them actually have a good level of purity in terms of the aggregates level. For this one, over 45% of aggregates were present after affinity capture step. So we utilize still 3-step chromatography. For this particular case with AEX and CEX optimization, successfully remove all of the impurities and achieve greater than 99% of the SEC purity toward the end of the process. So our last topic for today is on the N-glycan optimization. Over the recent years, we have seen more and more cases and clients asking for the glycan profile optimization because over the years we have accumulated more understanding about how does glycan profile impact different properties of our target IgG molecules. For example, these are a number of commonly seen glycan profile that's with their potential impact on antibody are pretty well understood. For example, the high mannose species could have impact on the serum half-life, and oftentimes we have to control the mannose level down to at least within 10%, for a lot of cases within 5%. And afucosylation, we know that some of the cases we need 100% afucosylation to successfully enhance the ADCC effect. And glycosylation could have certain impact on the CDC as well as the ADCC, and cellulation we have seen it also has impact on the ADCC as well as its half-life. So how to properly control the glycan profile so that we can achieve the desirable glycan profiling? To address this problem, let's first go over how does the different glycan profile was formed inside the CHO cells. So basically, the glycan profile typically happens in Golgi or the refolding and modification process. So in general, this process involves a number of enzymatic reactions that involves both of its precursors as well as a number of enzymes involved in this reaction chains. So in general, in order to manipulate a desirable N-glycan profile, we can adopt the general strategy either through the cell line engineering by tuning certain mechanism and their kinetics, and through the cell line engineering, or through controlling the cell culture conditions to adjust enzymatic reaction, or adjusting the precursor or its competitor's concentration. Let me further illustrate through a couple of examples. In our platform, we have done quite a number of efforts in terms of cell line engineering to achieve a target glycan profile. So first, with our proprietary afucosylation cell line with ADCC plus functionality, this is with FUT8 gene knockout. This is a quite well-known mechanism to achieve ADCC plus functionality, and we can basically achieve 100% afucosylation through this proprietary cell line. And in addition, we have seen emergent applications in high stylation profile as well as reduced mannose profile, so that we have also designed in-house our proprietary cell lines that could produce reduced level of high mannose as well as increased level of stylation. This high stylation cell line is going to be launched soon by next year. And in terms of controlling the cell culture conditions, as I have mentioned, the two general ideas is first to control the enzyme activity. For example, a lot of cell culture parameters could have certain impact on the enzyme activity, with most commonly used ones including the pH. Sometimes it's the media, the osmol level, cell culture duration, and DO level could also impact the enzyme activity. And alternatively, we can control the concentration of the sugar nucleotides, which is a precursor of the glycan species, with their tuning their precursor cofactors or analogous concentration to achieve either improve a certain level of glycans or reduce the level of glycans, with some commonly used parameters summarized in this table. Here I give a couple of examples in real projects that we have adopted in the previous slide strategies. By tuning the cell culture parameters, we can achieve either improved glycosylation level, improve the afucosylation. Again, this is not as effective as using ADCC plus cell line, but at least we can see somehow a significant increase of afucosylation percentage. How to control mannose 5 species, as well as improve the stylation level. So in summary, from this talk, hopefully I have demonstrated that we have quite extensive experience with complex biologics, including bispecific antibodies as well as fusion proteins. And in the last, I would like to also highlight that we have successfully assisted a number of our partners in completing a number 6 licensing out projects, as listed details here, which covering bispecific antibodies, fusion proteins, and ADC formats. And some of these molecules has been successfully licensed out to global MNCs, and over the past 2 years, we have been continuing partnering with our company friends to continue providing the we have successfully provided IND enabling CMC service and continued providing the material production for their clinical phase use. And in the last, we recently also launched our Express CMC service with the industry-leading development speed. As I mentioned, we have our proprietary ProMax vector launched earlier this year, and together with this vector that allows us to deliver this 4 months tox material delivery coming from a single clone material. To our knowledge, this is one of the fastest industry-leading development speed, utilizing a single clone to produce the tox material. And we are in the process of promoting this service and going to give you further details in September through the webinar series. And you are more than welcome to stay tuned for this further introduction on this new service. With that, that brings to the end of my talk, and welcome any questions.
혹시 질문 있으시고 편하게 질문해 주시겠습니까?
네. 어플리케이션을 방지하기 위한 방법으로 링커를 늘리셨는데, 링커를 늘리고 나서 다이설파인 본드가 원활히 형성된 이유가 무엇인지 궁금합니다.
Could you go back the aggregation part in?
Yep. Which slide?
I think they're disulfide bond. Yeah. So she wonders we modify type for the for disulfide bond.
근데 다이살파이브가 원활히 형성된 이유가 뭐지? 링커를 돌린 이후에 링커가 지금 세 개, 그러니까 GGGS 세 개?
네네네.
넣었었는데 지금 3 플러스 N으로 되어 있으니까 그 이상을 보시고 나서 다이살파이드 본드가 좀
네네.
원활히 이루어져서 어그리게이션이 방지된 것으로 보이는데
네네.
어떤 원리를 디자인을
Okay.
하셨나요?
The compare the wild type and the modified type, so we increase of the linker. So the what is the some reason to make a disulfide bond for here?
That's a good question. So, a lot of times when we see the client like to utilize single traffic because it's the easiest thing. You can take all the binding site from the wild type IgG and then utilize it in your bispecific antibody formation. But sometimes we see it's not a very good idea in terms of the CMC perspective, because in their wild type, in the IgG structures, actually all the hydrophobic patches are closely packed inside the IgG antibody because of their nature formats. But if you simply take out the single strand free fragment, and you have to find some ways to basically connect these two different domains, and making sure that, because there are a lot of hydrophobic regions inside the single traffic, and without a proper design, it's very easy to expose certain hydrophobic patches and make them easy to form aggregates. So in this case, a commonly used strategy is to introduce a linker, and with tuning different lengths, as well as interchain disulfide bond. Basically, the idea is to stabilize the two species and minimize exposure of hydrophobic region. Because otherwise, if you let these two species freely form, it's very likely to introduce a lot of hydrophobic patches exposed to form certain aggregates. So in our experience, the first introduction is always to introduce interchain disulfide bond, but with respect to a specific spot of mutation, that probably needs to be further optimized depending on different cases. And alternatively, with the addition of disulfide bond, sometimes we can also consider adjusting the length of this interchain linker to basically alleviate certain conformational stress or improve its thermal stability. I hope that helped address your question.
말씀 주신 것처럼 지금 와일드타입 같은 경우는 하이드로포빅이 많기 때문에 어그리게이션이 심하게 많이 지기 때문에 지금 다이설파이드 본드를 하거나 링커를 늘림으로써 이렇게 방지를 했다, 이렇게 설명을 해주셨습니다. 혹시 추가 질문 또 있으실까요?
네, 없으시다면 두 번째 섹션 여기서 마치도록 하겠습니다. 혹시라도 궁금하신 사항 있으면은 이제 두 번째 세션 끝나고 브레이킹 타임이 시작될 거니까 그때 저희 쪽으로 문의 주시면 될 것 같습니다. 네, 감사합니다. 땡큐인. 네, 전반 두 섹션이 지금 종료되었고요. 좀 유익하신 시간이 되셨으면 좋겠습니다. 지금부터 저희 15시 40분까지 브레이킹 타임 및 네트워크 타임을 좀 가지려고 합니다. 저희가 네트워크 타임 동안 좀 편하게 즐기실 수 있도록 다가를 좀 준비했어요. 저희 등록 데스크가 위치한 복도에 다가가 준비되어 있으니까 편하게 드시면서 네트워크하고, 혹시라도 업무가 있으신 분들은 편하게 업무하시는 시간이 되셨으면 좋겠고요. 저희 들어올 때 럭키 드로우 이벤트 혹시 참여하지 않으신 분은 명함만 넣으면 간단하게 이제 참여하실 수 있으니까 꼭 참여 부탁드리고, 섹션 종료 전에 부득이하게 퇴장하셔야 되는 분이 계시다면은 주차 등록 잊지 마시고 저희 설문조사 미리 해서 보여드리면은 기념품 먼저 드릴 수 있으니까요. 이 두 개 잊지 말고 챙겨가시길 바라겠습니다. 네, 다음 섹션은 3시 40분부터 시작되고 시작 10분 전에 다시 한번 안내드리겠습니다. 즐거운 시간 되시기 바랍니다. 네, 시간이 다 돼서 세 번째 섹션 시작하도록 하겠습니다. 네, 세 번째 섹션의 주제는 기네요. Integrated Single Domain Antibody Discovery via Phage Library and Single B-cell Screening Technology라는 주제로 첫 번째 섹션을 담당하신 대릭님께서 다시 한번 연사를 맞아주실 예정입니다. 박수로 환영 부탁드리겠습니다.
Welcome back. So the first topic for the second session is about the single domain antibody discoveries using different platform technologies. So comparing to the traditional monoclonal IgG or the IgG fragment like a Fab or single chain FVs, single domain antibody comes with a much smaller size and better tissue accessibilities. And the single chain format gives it a unique flexibilities in engineering to fit different kinds of formats. And comparing to the single chain FVs, the single domain or the VHH is generated in vivo by animal immunizations. So it has a pretty decent developabilities and manufacturabilities. And most of all, the single domain antibodies has been validated in terms of the efficacies and the safeties by a lot of clinical pipelines. Over the last decade, the single domain antibody is widely used in bispecific or multi-specific antibodies as a building block, or in cell therapies like CAR Ts. In recent years, the single domain antibodies is gaining more and more interest in gene therapies, especially in the in vivo target delivery for the virus or for the mRNAs. So this is a summary of some high-profile single-domain antibody-based therapies. So far, there are 4 approved single-domain antibody-based therapeutic antibodies for the indications like oncologies and autoimmune diseases. There are more than 140 pipelines globally. Most of them are in the preclinical or early clinical stage. ProBio offers 3 different approach to generate single domain antibodies. Both the immunized and the naive alpaca libraries are built based on the phage display technologies. Alternatively, we can also screening for single domain antibodies at single B cell level using the Beacon platforms. We will share more detailed technical information in the next couple of slides with these 3 different strategies. After recovering candidates, these leads will be further characterized by in vitro assays, followed by engineering such as humanization affinity maturations and in vivo animal disease models, and also the developability assessments. It's a pretty standard process. So these 3 different platforms to generate a single domain antibodies has its unique features, and it comes with its specific application scenarios. For instance, if your top priority is really good diversity, and the timeline is also really sensitive, so people usually will go with the Beacon-based single B cell strategies. Alternatively, the immunized phage display libraries can generate antibodies with higher affinities, but when your target antigen has a really high homology to the camelid animals, it may be potential issues because of the immune tolerance. Under that circumstances, we can resort to the naive library. Without the immunization, you can so basically you can eliminate the immune tolerance issues, and you can generate the antibody leads using the ready-to-use naive library. But one of the main issue for antibody coming out of the naive library is the affinities. Comparing to the other 2 approach, the antibody affinities is somewhere around 10 to the -7 to -9. roughly 100-fold lower or worse than these two immunized-based approach, both single B-cell approach and the immunized approach. And this gives you a general idea about the workflow for each of these different strategies. For single B-cell based strategies, we start with the animal immunization. After recovering and enrich plasma B-cell from the PBMCs, these B-cells will be loaded onto the chip for single B-cell antigen binding screenings. The positive B-cell will be exported for recombinant expression and confirmation by antigen binding affinity and cell-based functional assays. And after the functional confirmation, the top candidate will be sequenced for scale-up production and in vivo validations. Alternatively, for the immunized fetal display approach, we also start with immunization. The PBMC will be licensed to recover the mRNA and to build the phage libraries. Usually the library will be panned and screened by different kinds of antigens, including soluble proteins and recombinant cell lines or any other antigens of interest, followed by candidate sequencing, recombinant production, and functional validations. The naive library workflow is pretty much the same as the immunized library minus the immunization and the library construction part. So the naive library takes much less time with the sacrifice of antibody affinities. So for the single B-cell approach, there are 3 key steps. The first key step is to enrich for the plasma B-cells. When we were doing this kind of project, first we will use the antigen to enrich for the antigen positive memory B cell. And the memory B cell will be converted into plasma B cell in vitro by proper stimulations. And these plasma B cell will be loaded on chip for the screenings. The key feature of this Beacon platform is it is able to run multiple runs of antigen binding on the chip so that you can successfully narrow down your candidates from thousands of positive B cells to probably 50 to 200 positive B cells. And even with, let's say, 100 positive B cells, the traditional approach following the B cell screening is to do the sequencing, gene synthesis, plasmid preparation, and recombinant production. It's gonna be a huge amount of work to do the whole process with 100 or 200 positive B cells. So that's why we have the third key step, the ProSpeed expression platform. So instead of doing sequencing and the gene synthesis and the plasmid preparations, we will do the PCR with the positive B cell to clone out the antibody fragment, the variable region fragment, and clone it into a linear plasmid by homology recombinations. It's much convenient, much more easier than the traditional gene synthesis and the ligation and the PCR reactions. Then we can take this reaction mixture directly for transient transfection with mammalian cell lines like CHOK1 or HEC293s. It's exactly the same as a traditional circular plasmid-based recombinant production. And after this recombinant production, we will harvest the supernatant for binding and the functional assays, cell-based functional assays. So with this ProSpeed expression, we're able to handle hundreds of positive B cells with a relatively low cost and a relatively shorter timeline. Besides the single B cell the phage display also provides a alternative option for the single domain antibody discoveries, including both the naive library and the immunized library, as I mentioned in the previous slides. So in addition to the library itself, the panning strategy also plays a very crucial role in the phage display-based antibody discovery strategies. Here is a list of the most common panning strategies we often use nowadays. The liquid panning is basically approach to capture the antibodies by biotinylated antigens in the suspensions, while the solid antigen panning is to capture the antibody by the antigen immobilized on the plates. The cell panning is basically using engineered cell or cancer cell to capture the antibodies. For any project, we will typically combine different kind of panning strategies to improve the overall success rate and to maximize the diversity of the epitope and the diversity of antibody sequence. Traditional phage vector, phage plasmids, the antibody is actually fused to the P3 proteins of the phage. So that's how the antibody is displayed on the surface of the phage. But that also brings a major issue. These phage will significantly interfere the antibody antigen binding. So that's why you often see non-specific binding background signals in ELISA or flow cytometry assays when you use the phage supernatant. So that's why when we use this phage vector, it comes with a AMBER stop codons. When we induce the cell with IPTG, the expression of the antibody will stop at the AMBER stop codon as a soluble version without the P3 proteins and the phage. So this soluble version of antibody candidates will greatly reduce the signal noise, the non-specific background in ELISA and the flow cytometry analysis. And also it makes it possible to run the SPR affinity ranking with the phage supernatant. So after generating the top candidates, the next step is engineering. The humanization of a single domain antibody is pretty similar to the humanization of like rodents or rabbit antibodies. It's still based on the CDR grafting and framework back mutation strategies. The only differences is there are several conserved residues in the framework 2 of a VHH. So there are certain special design for the back mutation when you consider doing the humanization of VHH single domain antibodies. Over the last decade, our success rate of a single domain antibody humanization is roughly at 91% plus 5%, so that's 96%. As I mentioned previously, if the antibody is generated from the naive library, the affinities might not be very good. So in that scenario, we may need to do affinity maturations to in vitro affinity maturation to improve the affinities. So ProBio offers a pretty unique strategy for affinity maturation. It's called two-step library-based approach. In the first step, we will generate a saturation mutation library for each critical residue in CDRs. Let's say if you have 30 critical residue in the CDR, we will generate 30 saturation mutation libraries. And we will go through these saturation mutation libraries with SPR affinity ranking, and those beneficial mutation, the one can improve the affinities, will be isolated and built into a secondary library called combinatorial mutagenesis libraries. And after further affinity ranking with the combinatorial mutagenesis libraries, we will end up with the top candidates with the most affinity improvement. Once the sequence is finalized, the next step is the developability assessment. Actually, there is 3 different checkpoints for developability assessment. The first checkpoint is at the time when you do the humanization. Usually we will include immunogenicity predictions, post-translational modification analysis, and aggregation analysis along with humanization process. And the second checkpoint is after the sequence engineering. It's a cell-based assays. The primary purpose of the checkpoint 2, the in vitro developability assay, is to evaluate the non-specific bindings, the self-interaction, and the stabilities in plasma, basically in ex vivo stabilities. And in the third checkpoint, which is roughly the time when you do the in vivo animal studies, we will do the manufacturability assessment. The purpose of this step is to evaluate the stabilities of your candidates under stressful conditions like high temperatures, repeated freeze-thaw cycles, and acidic or basic conditions. So that's a general introduction of the overall workflow for single-domain antibody discovery and engineering. And the next, we will share 3 case studies corresponding to each of those platforms I mentioned at the very beginning. So the first case study is the discovery of a GPRC5D leads for T-cell engager bispecific antibody using the alpaca immunized libraries. We already talked about the nature of the target, the GPRC5D, at the first talk, so I will probably skip this information. So similar to that ADC case studies I shared in the first talk, we also use the mRNA to immunize the alpaca to generate the immunized phage libraries. And the library, similar to that hybridoma approach, the library is paired and screening against the engineered overexpression cell line and the myeloma cell lines. After confirmation by flow cytometry bindings and the sequencing, we end up with top 12 candidates. And we select 6 top candidates from this top 12 for T-cell engager bispecific construction and the T-cell activation RGA assays to evaluate the activities of the T-cell activation with these T-cell engager bispecifics. So similar to the previous hybridoma case studies, for this alpaca immunization projects, we also measure the cell titer data using both engineered cell line and the myeloma cancer cell lines. And we also measure the IgGs and the VHH titers. It is a crucial step to measure the VHH titer because most of the camelid animals is able to generate both IgG antibodies and the VHH antibodies simultaneously. And the percentage of VHH antibodies is usually around 30%, but it varies from animals to animals. So that's why it's crucial to make the decision based on the VHH titer, but not solely on the IgG titer, because the VHH is actually the one we need. After generating the libraries, the library will be subject to panning using the antigen cell lines for multiple runs. In this case study, we use the engineered overexpression, high expression cell line for the first round of a panning, and use the myeloma low expression cell line for the second round of pannings. After the pannings, we generate 12 top candidates, and we start the more detailed characterizations with these top 12 candidates. The first, again, it's the flow cytometry-based antibody affinities against the myeloma cell lines and the engineered cell lines. in both studies, these top 12 candidates showed equivalent or better cell binding affinities comparing to the clinical benchmark. Based on that affinity data and the sequence cluster analysis, we selected a few top candidates to generate 6 T-cell engager bispecific antibodies with 1 CD3 antibodies. And these 6 bispecific antibodies showed various activation level of the T-cell by these RGA assays. And when we treat these bispecific antibodies with antigen negative cell lines, there is a minimal level of a T-cell activations, confirming that these bispecific antibodies has a minimal non-specific spontaneous activations. It is a pretty crucial indicator for the safety and efficacy of a bispecific CD3-based T-cell engager bispecific antibody. Okay, so that's for the first case study for the immunized libraries. The second case study is the discovery of ROR1 antibody for CAR T cell therapies using the naive libraries. ROR1 is another popular tumor-associated antigen in recent years. It's widely used for T-cell engagers, ADCs, and CAR T-cell therapies. One of the major issues for ROR1 is it has extremely high homology between different species, human, cyno, mouse, and alpacas. So in this particular scenario, if you immunize the animal with ROR1 proteins, most likely you will not generate any immune response because of the immune tolerance issues. So that's why we decided to use the naive library to avoid the immunization issues. So after panning and screening through this naive library, we actually generated more than 400 hits. And based on the sequence cluster information and the relative affinity rankings, we chose 30 of them to move forward for single-chain antibody recombinant production and purification and further characterizations. And based on this data, we selected 8 top candidates which targeted to each of these 3 extracellular domain of ROR1. Next, we take these 8 unique candidates for the CAR construction using this particular structure on the right panels. And we further evaluated the efficacy of the CAR using these 8 different antibodies. The first experiment we did is a luciferase-based killing assays. It's a pretty common assays to measure the efficacy of CAR T cells. From this experiment, we identified top 3 candidates showing equivalent or comparable killing efficacies to the benchmark, the R12. So in addition to the luciferase assays, we also measured the activation of the CAR Ts by the induction level of IL-2 in the middles and interferon gamma on the right panels. Again, the same 3 top candidates gives equivalent or comparable induction level of these cytokines. And luckily, these 3 top candidates accidentally targeted to each of these extracellular domains, so that we chose these 3 top candidates to move forward for humanizations. So each of these candidates were successfully humanized, and the affinity is successfully maintained at 10 to the minus 7 level. Remember, I mentioned that the antibody affinity come out of the naive library is usually worse than the immunized libraries. So that's why we chose one of the candidates to do a further affinity maturation. Affinity is improved from 10 to the minus 7 to 10 to the minus 9. And we use that affinity mature the variant to get a further confirmation by the in vitro CAR-based killing and efficacy assays. So that's the second case studies using the naive libraries. The last case study is the discovery of antibodies against a immune checkpoint target using the beacon-based single B-cell approach. As I mentioned at the very beginning, the overall process of the single B-cell based approach is like start with the alpaca immunization followed by plasma B-cell enrichment, B-cell screening on the chip, followed by pro-speed expression, validation, and sequencing. The whole process takes about 3 months. Again, during the immunization stage, we monitored the serum titration data again using specific secondary antibodies, either against the total IgG or the IgG2 and 3. The IgG2 and 3 represent the VHH. We also measured the serum titration data against both human and cyno-monkey proteins for the verification of monkey cross-binding affinities. Then we harvested the PBMC from the selected alpacas after 5th and 6th immunizations. And after the enrichment of the plasma B cells, we started the B cell screenings. So for the sample after the 5th immunizations, we identified 156 positive B cells, and 151 of them is successfully PCR'd. The success rate of the PCR is roughly at almost 97%. And for the second batch of the samples after the 6 immunizations, the PCR success rate is also roughly at 95%. And 100% of these PCR fragment is actually confirmed as a VHH, but not IgG. This is a very crucial step because we do not want to end up with a bunch of candidates that is a full-size IgG, but not VHH. After recombinant confirmation, 151 and 179 of the recombinant samples are actually confirmed as a positive binder to the recombinant antigens. And based on the sequence analysis, 58 and 55 of them are the unique sequences. So all of these are pretty crucial indicators we constantly monitor during the B-cell screening stage. And this recovery rate is considered pretty high because the industrial average level of a recovery rate from positive single B cell to confirmed sequence is roughly at 30% to 50%. So after generating this top candidates, we select a few of them for further characterization, starting with binding affinities against recombinant protein using ELISA, or against the overexpression cell lines using the flow cytometries. So as you can see from here, the majority of these candidates showed relatively higher binding affinities in both ELISA and the flow cytometries comparing to these two clinical benchmark. In addition to the antigen bindings, since this is an immune checkpoint target, we also look at the receptor ligand blocking capabilities with these candidates, some of these candidates. And judging by the ELISA-based competition assays, we can conclude that these leads showed a differentiated profile of blockings. Some of them showed really good blocking to the receptor and the ligand binding. Some of them do not. So this further suggests a really good, the diversity is well maintained after the whole screening process. And in the last step, we put all of this sequence together to do a phylogenetic phylogenetic tree analysis because there are too many sequences, so I just put some of them here. So the yellow highlighted ones are the sequence recovered from the 5th immunizations, and the blue highlighted ones are the sequence recovered from the 6th immunizations. So the sequence recovered from these two independent batch of samples shared a even distribution within different germline of the antibodies, suggesting really good diversities can be recovered from this single B cell based antibody screenings. Okay, so that's all for the talk. Any questions?
질문 있으신 분들은 자리에 손을 들어주시면 저희가 마이크를 건네드리도록 하겠습니다. 네, 혹시 지금 시간 이후라도 질문 있으시면은 저희 스태프분들 통해서 개별적으로 질문 부탁드리겠습니다. 네, 땡큐 자리.
Thank you.
네, 오늘의 마지막 섹션입니다. 마지막 섹션은 다시 한번 이익님께서 프로바이오 플랫폼 솔루션 for VHHN 안티바디 프래그먼츠라는 주제로 발표를 해주실 예정입니다. 네, 큰 박수로 이익님을 환영 부탁드리겠습니다.
All right. So let's further continue our discussion on the VHH and antibody fragments. So for this talk, our focus would be the CMC solution for this different type of antibody fragments. Here is the contents to be covered in this talk. We'll start with some background and application of antibody fragments, followed by the CMC strategy for different type of antibody fragments, including single chain FV, Fab fragment, as well as the single domain antibody, along with relevant case studies from our platform. So here is a general overview of different applications and advantage of antibody fragments. So as Derek mentioned in his talk, antibody fragments, different from the full-size IgG molecule, they are small molecular weight, which allows for stronger tissue penetration and enables improved permeability through solid tumors and tissue barriers. And also their simply structure allows for flexible design, which is ideal for engineering for different type of bispecifics, multivalent, or other advanced formats. Last but not least, the small size of antibody fragments are well suited for conjugation applications. In particular, over the last year, we have seen a booming type of applications utilizing different antibody fragments for conjugate applications. In particular, we have seen an increase in use of antibody fragments in in vivo CAR T cell therapies. Basically, the ideal of in vivo CAR T is to deliver the CAR gene utilizing the mRNA LNP structure with the targeted purpose for the antibody and to be delivered in vivo in order to avoid the complex ex vivo treatment and engineering of the CAR cells. So to synthesize the antibody-targeted lipid nanoparticles, there are different general approaches. We could either utilize a one-part type of synthesis, basically putting all the components, including the antibody, mRNA, as well as the lipids all together and through a one-stop synthesis. Or we can do form the LNP mRNA first and do the surface conjugation utilizing the antibody. And the last approach that's commonly seen is the post-insertion approach. So we can conjugate the antibody fragment to the lipid and then form the LNP particles later. So in general, this platform has been established by our mRNA CMC team along with the QC and analytical platform. So for this particular talk, we are going to be focusing on the antibody part for the antibody LNP applications, including different type of antibody fragments, such as a single domain antibody VHH, which so far we have seen are the most commonly used type, along with single chain FV fragment as well as Fab fragment. So first, let's start with the expression system of different antibody fragments. In general, depending on different complexity of these molecules, we can either utilize the CHO cell expression versus the E. coli cell expression. So in general, the E. coli expression allows for a much shorter construction cycle within 2 to 4 weeks, and the expression level could be competitive depending on which type of molecule. For example, if that's a single domain antibody, the titer could be pretty high. And however, there are a number of advantages utilizing CHO cell over the E. coli system. For example, in general, we can secrete the protein utilizing CHO versus a complex lysis process because the protein expressed in the inclusion bodies typically for the E. coli system. And we have to establish a cell bank, including master cell bank and working cell bank, which complies better with regulatory standard requirement compared with E. coli without this establishment of stable cells. And that could also face, when it scale up, face some consistency issue as we scale up the process. And in terms of the product quality, for the CHO-expressed antibody fragments, there's no refolding needed, and it could facilitate certain post-translational modification as well. However, for E. coli, it typically requires the refolding process, and also along with higher level of HCP and endotoxin contamination issues. So in general, the CHO cells enables better GMP compliance as well as regulatory compliance and good quality consistency across different batches as well as scale up. Hence, this is a recommended approach for our platform for the production of different antibody fragments. So next, I will go through different strategies for different type of antibody fragments, starting with the Fab fragment. So for the Fab fragment, the typical challenges we have seen during the expression are the sometimes lower expression level compared with IgG. And a lot of times, imbalanced chain expression. So we sometimes see overexpression, for example, the light chain, hence the formation of some homodimer species and limited protein stability. Therefore, we optimize our upstream expression platform, utilizing a dual vector design and optimization. Keep in mind, during the early stage cell cloning, for high assembly of efficiency and optimize upstream feed and temperature control strategy to maximize its expression level. For the downstream purification, because of these molecules' lack of Fc structure, we have to come up with a dedicated affinity platform to capture these antibody fragments. And again, sometimes we see aggregates and homodimer along with fragments, impurity present in the cell culture that need to be effectively removed in the downstream process. Additionally, we have some challenges in the analytical development to target specifically for this type of molecules and identify the impurity that are present in the cell culture. So here I'm going to walk you through a case study for a Fab molecule. This molecule we have filed for IND for FDA in the past year, probably 2 years ago. So in general, we haven't seen too much challenge in the upstream. So utilizing our platform process with optimization, the expression titer could reach close to 8 grams per liter. And there are a number of challenges, mostly around downstream and analytical side. So first, as I mentioned, we have to come up with a good capture step without the Fc fragment. And in general, the binding capacity of this molecule on selected resin could be very low. And last but not least, the presence of high level of aggregates and homodimer needs to be removed. So let's start with the selection of the affinity resin. So we systematically screened different type of resins targeting different binding domains. For example, for Fab, we could look for resins that's targeting VL. That's typically protein L, protein L are the most commonly ones that you could think of. Or targeting VH, actually, protein A has sometimes good affinity with the VH region. And there are also affinity resins designed to capture the CL and CH1 region, respectively. So in this project and overall in this program for the Fab molecules, we actually screen different type of affinity resins, and at the end of the day, identified the optimal resin for this project targeting the VL region, and with much higher binding capacity compared with the rest of the resins, therefore successfully reduce the overall production cost for the downstream. And further utilizing this affinity resin, we developed effective method using the elution strategy to remove the majority of aggregates and homodimers, as shown in this slide. So after we pick up the affinity resin, we did a linear gradient elution at different pH and try to see if there's any resolution of our main protein of interest versus the rest of the impurities. As I mentioned, actually, interestingly for this molecule, the main impurity species are the homodimer. And there are two different types of homodimers, either formed covalently or non-covalently, as we will see from the analytical section later. So during the affinity optimization, we find different elution pH has a significant impact in separating different impurity with our target antibody. Therefore, we selected the optimal elution pH and successfully separate those impurities utilizing isocratic elution. As you can see that through the AC step, we can achieve close to 100% of the ICC purity with a decent recovery rate over 80%. And we further have two polishing steps to remove the additional impurities such as HCP and HCD. And the overall recovery is at 72%. Two additional points to be highlighted for this molecule. You can see graph on the left-hand side. This molecule is kind of special in the sense that it requires very high SEC purity in order to prevent the toxicity of this molecule. As you can see from this graph, the ELISA binding activity correlates strongly with the high molecular species. As you can see, if we have a high molecular weight species greater at a very low level, even at less than 1%, there's a significant increase in the ELISA binding activity. And this enhanced ELISA binding activity is kind of correlated to the toxic aspect of this molecule. So this brings up very challenging CMC aspects in the sense that we have to tightly control the aggregates level in order to minimize the toxicology of this molecule. So at the end of the day, actually the client asks us to control the ICC level purity to be above 99.5% and apply that in all the GMP production, and we actually did it. So we have actually successfully come up with a robust process that gives the ICC purity above 99.5% and tightly control the aggregates level, and successfully delivered the GMP production for a number of batches. And another interesting aspect about this project is how to quantify the different impurities. So in this case, as I mentioned, the major impurity are the overexpressed lectin homodimers. So actually, at the end of the day, we find out that CESDS-NR, non-reduced condition, is a good way to quantify the different species, because as we isolate the light chain impurities as around the SDS-NR. And as you can see that interestingly, the majority of the species, only a small portion belongs to the LC-LC species, which is formed by the disulfide bond. The majority of the LC-LC impurities are actually being break down at the NR condition because they are non-covalent bonded. So we come to the conclusion and that the majority of the impurity present in this project are the non-covalent bonded light chain homodimer species. Next, let's go through the general strategy for single chain FV fragment. I think we briefly touched base on the first talk, and I will go into probably give more details on the designing of the single chain FV in this part. So the single chain FV molecule fragment, when expressed by itself, whether expressed by itself or combination with IgG to form bispecifics, the number one challenge is always a high level of aggregates that's been present. And also this type of molecule has limited protein stability, meaning that it's prone to form more aggregates at certain pH or ionic conditions. And in some projects, we have seen low expression level for single chain FV compared with other fragments and antibodies, and sometimes low cell viability during the cell culture stage. So in terms of upstream strategies, similarly, we typically start with trying to optimize the design of the molecule in the beginning, and then choose the optimal clone for high monomer production. And keep the purity in mind in the early stage screening. And by optimizing the feed and cell culture temperature, that could further improve the overall performance of our cell culture. And in terms of the purification, similarly, there's no FC fracture. We have to come up with a good way to capture the single chain FV fragment. Prone to form aggregates, effective solution to remove those aggregates, as well as the poor stability of this type of molecule. So the solutions include screening a suitable capture resin, as well as along with a buffer system and elution condition and additives, utilizing multimodal or mixed mode chromatography to remove aggregates and other aggregate species, and control the protein concentration to avoid the degraded DS quality. So here is one example of a single chain FV fragment. So as I mentioned, in general, our platform has seen a lower expression level for single chain FV. For this case, it's 1.6 gram per liter through the cell line process. And during the cell culture, we have identified that the cell viability as well as the productivity of this molecule is heavily impacted by the cell culture temperature. So as you can see, we designed different temperature downshift at 30 versus 33 with 37 degree, and you can see lower temperature at 30 degree significantly help to extend the cell culture duration and save the viability drop. As a result, the titer as well as the QP of the molecule of the cell all improved by 6 times by modulating the different cell culture temperature. And we also considered extend further culture duration from 14 days to 17 days to further improve the expression level. And moving on to the downstream, we designed a protein L capture step to successfully capture this single chain FV fragment and choose the optimal mixed mode chromatography condition to effectively remove the aggregates. And in this project particularly, it actually involved multiple conjugation steps afterwards, utilizing a C-terminal free cysteine. And we systematically also optimized the conjugation conditions, including the feed ratio, the antibody concentration, reaction pH, etc. And at the end of the day, remove the aggregates by optimizing the AEX and mixed mode chromatography steps. At the end of the day, we achieve a high good product purity over 98% for both SCC and CE, and with overall recovery rate at 65%. So here is another example of the importance of design of single-chain FV component that will impact the stability and developability of a drug. So this case is also a 3-chain asymmetric bispecific antibody format. So initially, the client used certain design of a single-chain FV, which yields in a relatively low titer, as well as a high level of high molecular weights. So to deal with this challenge, I think our antibody engineering team also worked closely with the client to modify the design of the single-chain FV, basically by switching the VH and VL domain. We can see the titer increased significantly by 100% from 2 grams per liter to 5 grams per liter, and with a much better improved overall purity of greater than 90%. And this example we have discussed in the previous talk, the introduction of interchain disulfide bond could also have some impact on the stability of single-chain FV fragment. So next, and going to be the focus of this talk, is on the VHH fragment. So here is a comparison of the VHH from Camlid versus the human VH structure. So they are similar, but in terms of consisting of 4 frame regions as well as 3 CDR regions. So the main difference between the two different VH versus the VHH molecule lies in first that the VH has 4 highly conserved hydrophobic amino acids in their frame 2 region. And in the VHH, these 4 amino acids are being replaced by hydrophilic amino acids, therefore improve the solubility and stability of the VHH molecule. And in addition, the CDR1 and CDR3, there was introduction of interdomain disulfide bond, which further enhanced the structural stability of VHH compared with human VH. That's why we normally see quite good drug developability utilizing the VHH fragment. So in terms of its CMC challenge, for VHH molecules, sometimes we see lower expression titer, and for some molecules, for a number of VHH tandem format, we have seen lower cell viability. And sometimes the stability, even though VHH has good stability, but VHH combined with other components could yield in a low stability type of molecule format. Therefore, the upstream strategies are kind of comparable to the other counterparts. And for the purification, I'd like to highlight that in addition to, again, no FC structure, we come up with actually a number of different strategies designed for different type of VHH molecules. So we work with a number of different VHH molecule designs. So first one is kind of native VHH. Sometimes it belongs to the VH3 group. So the VH3 VHH actually has quite good affinity to the protein A molecule, protein A ligand, so that we can utilize protein A for capture. The rest of the VHH groups typically don't have any affinity to protein A, so that we have to come up with alternative solution to capture this group of VH. And for further conjugation applications, we have seen different VHH designs sometimes come with unpaired cysteines in their C terminal, so that allows for further conjugation to the LNP or the lipids or other different molecules. And sometimes alternatively, it could be an oligopeptide that allows for a site-specific conjugation using a certain affinity peptide series on the C-terminal. And other than the capture challenge, we've seen sometimes HCP removal as well as the formation of dimer, especially when this VHH is designed with unpaired cysteine on their terminal for conjugation. It's not uncommon to see a presence of high level of dimer present. All right, so next we'll go through a number of case studies to tackle the CMC solution for different type of VHH formats. So first group is the VHH with protein A affinity, which is a VH3 group. So this type of molecule is kind of straightforward. So we can utilize protein A. For example, the Prism A resin is commonly used with good affinity with VHH resin as a capture step. And in our platform, we have done a number of cases with just two-step chromatography to successfully reduce the residual HCP level down to within 100 ppm and with SEC purity greater than 99%. And just bear in mind, because of the small molecular weight, we have to utilize a small molecular weight cutoff ultrafiltration membrane for the UF-DF buffer exchange process. So the second group of molecule also is VHH with protein A affinity. However, this times they have the unpaired cysteines in their design for the conjugation purpose. So here is another example. For the upstream, we can achieve 6.5 gram per liter in their titer. However, the presence of these unpaired cysteines may lead to the formation of intermolecular disulfide bonds, hence forming the dimers, as you can see. Also from this gel, there's a significant portion of dimers present. So we work with different clients. There are different strategies to deal with this. First, you can certainly remove these dimers through adding the reductants. Typically with a low concentration of mild reductants such as cysteine could break down this type of dimers. Or alternatively, a lot of clients working on this type of applications, because their final application is to utilize this VHH is to conjugate. And the very beginning step of conjugation is the reduction step, utilizing strong reductants such as TSAP or DTT. So a lot of times they treat this dimer because we understand the mechanism of formation of aggregates pretty well. This dimer is due to the free cysteine formation. So we can also treat the dimer as our intermediate product. And upon the conjugation steps, by adding the reductants, this dimer could be successfully reduced. As we can see, we can either treat dimer as a final product for this DSI, or we can add a small amount of reductants such as cysteine to completely break down this dimer impurities. And in terms of the purification, since it has protein A affinity, we can potentially utilize protein A such as Prism A resin to capture the both monomer and dimer, but relatively low binding capacity and relatively expensive since affinity resin is involved. Alternatively, our platform has also developed our proprietary platform method, and we have in the process of our IP, is to utilize a mixed mode CEX to capture these VHH molecules, all utilizing CEX. So typically, either using CEX resin or mixed mode CEX, we can have a good resolutions between the monomers and dimers, and offering high capacity, binding capacity as well compared with affinity resin. However, if we utilize the mixed mode CEX or CEX as a capture step, we would typically expect higher HCP levels and needs to be further reduced, typically through three-step chromatography. And the third case the single domain antibody VHH without protein A affinity. As I mentioned, for this group of molecules, we have our platform method utilizing different CEX-based resin for capture. So here are a number of different cases for different clients, including just the VHH itself or VHH connected in series. So this is actually a bispecific antibody with different binders designed in this VHH tandem. And as you can see, through this platform approach, we can allow for very high SEC purity greater than 98% or 99% through the first capture step. And the problem is going to be HCP removal. So we designed a platform 3-step chromatography to deal with the HCP removal problem. And I think this is the last example. This is a VHH in series. So the titer is not bad at 5 grams per liter, but it took us quite a lot of efforts to get here, because initially when we do the cell pool screening, we saw this molecule, probably they are kind of difficult to be folded or expressed, and we've seen a very significant drop of cell viability at the medium screening stage. So we did a careful selection of the combination of different medium with respect to the different single clone, because we see clone and media compatibility could also impact the final condition selected and final viability of the process. And also, similar to the other projects, the cell culture temperature shift plays a significant role in the viability maintenance. As we can see, if we even different temperature shift time. This one we first determine the optimal temperature, and shifting the temperature at different days also have some impact on the viability. We cannot do it too early, but if we do it too late, it may impact our titer of impression. So this is basically a balance between the cell growth and the protein production. At the end of the day, we select the optimal temperature shift day at day 7 with the optimal downshift temperature. So moving on to the downstream, we're seeing this molecule is not quite stable, so we see a precipitation occurs during conductivity adjustment when we utilize the mixed mode CEX as capture step. And additionally, HCP removal has some certain challenge. So we did a number of steps, including adding intermediate depth filtration to remove the precipitate HCP through the conductivity adjustment, and add wash 2 in the polishing step to remove additional HCP species. And again, the molecule cannot bear with the low pH condition, so we designed solvent detergent treatment for this molecule, and we've seen low nanofiltration throughput during the nanofiltration stage, and we successfully identified certain metal ion that could, during the formulation development, could help to stabilize this molecule, and it's been added to improve the nanofiltration throughput. As you can see, before adding this additive, the nanofiltration throughput dropped very fast through the experiment, and after the adding metal ion, we successfully maintained a much higher throughput for the nanofiltration. At the end of the day, this project delivered over 99% of SEC purity along with the CE purity, with a controllable level of 20 ppm HCP, and with overall recovery of 70%. I would say this is overall challenging project, but we have successfully conducted the talks as well as GMP production, and in the process of, we have submitted IND for this particular case and waiting for the results. So in summary, here through this talk, I'd like to highlight that our platform has quite good experience working with different antibody fragments as well as the antibody or AXC. It's a shame that we don't work with ADC, but we work with a number of emerging AXC type of applications. So particularly, we work with a number of pretty well-known companies on the RDC, which is a radionuclide and antibody conjugates, which allows for targeted radio labeling type of treatment. As well, we are establishing our AOC platform, which stands for antibody oligonucleotide conjugation. This is also an emerging field for targeted oligonucleotide treatment. And along with the previous example I mentioned, the antibody LNP for in vivo CAR T type of therapy. So our platform covers end-to-end from the early phase discovery to developability study, to naked antibody process development and conjugation process development, formulation development, targeting for this different type of emergent AXC, so antibody plus non-toxic type of conjugates, along with analytical method development and GMP manufacturing for both naked antibody reagent conjugation manufacturing, as well as the fill and finish capability. So the last slide also shows our API service, because for a lot of these antibody fragments, they are not the final DP. So it's a kind of intermediate drug substance, which is going to be utilized for further conjugation to form the final drug modality. So we provide a much faster and more flexible type of service targeting this type of antibody fragments, which also complies with the regular requirement and for different type of applications, whether that's a GMP-like material or it's for the IIT type of application with a much faster turnaround time. So in general, we can deliver the IIT type of material within 3 to 4 months for this different type of antibody and antibody fragments, and 4 to 6 months to get the API materials that meet the IND filing requirements. With that brings to the end of my talk, and happy to hear if any questions or feedback. Thank you.
네, 질문 있으신 분은 자리에서 손을 들어주시면 저희가 마이크 전달해 드리도록 하겠습니다. 네, 질문이 따로 없으신 것 같아가지고 네, 여기까지 마치기로 하고요. 혹시라도 따로 질문이 있으시면은 저희 스태프분들께 요청 부탁드리도록 하겠습니다. 네, 감사합니다. 땡큐잉. 네, 오늘 이제 저희 프로바이오가 준비한 모든 섹션이 종료가 되었습니다. 네, 긴 시간 동안 좀 경청해주신 기민 여러분께 저희 다시 한번 감사 인사를 드리고요. 이제 모두가 기다리셨던 이제 럭키드로 추첨을 이제 시작하기 전에 네, 저희 이제 금일 심포지움 진행한 거에 대해서 여러분의 좀 귀중한 후기를 듣고자 합니다. 네, 이렇게 저희 앞에 보이시는 서베이 QR코드를 통해서 이제 서베이를 해주시면은 여러분의 소중한 의견을 바탕으로 내년에 준비될 심포지움은 더욱 발전되고 유익한 심포지움이 될 수 있도록 준비하겠습니다. 저희가 한 3분에서 5분 정도 이제 설문조사하는 시간을 갖고 럭키드로 추첨을 시작하도록 하겠습니다. 설문조사 완료하신 분들은 이제 화면 완전히 끄지 마시고 나갈 때 저희 스태프분들께 한번 보여주면은 기념품 전달 드릴 예정입니다. 네, 이제 설문조사가 어느 정도 다 끝난 것 같습니다. 네, 그러면 이제 가장 즐거운 시간을 이제 앞으로 갖도록 하겠습니다. 저희 3등 5분, 2등 3분, 1등 1분 이렇게 추첨을 진행할 거고요. 여기 보이는 이제 명함을 랜덤으로 뽑아서 추첨을 진행할 예정입니다. 3등 추첨은 저희 잉님께서 좀 도와주시겠습니다. 잉, could you please take 5 minutes card? Don't get the box. 네. 드노버 바이오 테라피틱스 박흥록 연구자님. 네, 축하드립니다. 네, 당첨되신 분들은 이따가 단체 사진 한번 찍을 예정이라서 잠깐만 단상에 기다려주세요. 네, 5분 다 부르고서 한 번에 드리도록 하겠습니다. 네, next. 네, 셀럽메디의 한기찬 선임연구원. 네, 축하드립니다. 네, 매드팩토 이창모 팀장님. 네, 축하드립니다. 네, 킴셀엔진의 김성찬 책임연구원. 다행히 모두 계시네요. 네, 마지막입니다. 네, 서울대학교 병원의 최정환 교수님. 네, 3등 당첨되신 분께는 저희 스타벅스 쿠폰. 네, 감사합니다. 잠깐만 기다려주시죠. 저희 2등 다 뽑고 단체 사진 한번 찍을 예정입니다. 네, 다음 2등은 대리님께서 뽑아주시기로 하겠습니다. 대리, please take 3 business card. 네, 매드팩토 김은주 이사님. 네, 매드팩토 김다은 과장님. 매드팩토 분들이 상품을 이렇게 들어가시는 거예요. 라스트. 네, 뉴라클 사이언스의 장현숙 상무님. 네, 2등 당첨되신 분들께는 스타벅스 텀블러 제공드리겠습니다. 그리고 저희 이제 마지막 1등만 남았습니다. 1등은 한 분밖에 안 남았는데 제가 한 가지 팁을 드리면은 제 명함 받아보신 분들 알겠지만 명함 두꺼워요. 네, 두꺼우면은 이렇게 손에 잘 걸리더라고요. 제가 올해 몇 번 걸려봐서 아는데 당첨 안 되신 분들은 회사 돌아가셔서 이제 명함 바꿔가지고 내년에 다시 한번 트라이 부탁드립니다. 네, 마지막 1등은 저희 한국 부문장이신 주문경 상무님께서 뽑아주시겠습니다.
네, 진호맨 컴퍼니의 이지영 선생님. 네, 축하드립니다.
자리 안 계신가요? 아, 다행이네요. 지금 몇몇 분들 얼굴에 이게 화내기도 왔던 것 같은데 그러면은 이제 금방 돌아오실 거라고 믿고 잠깐만 좀 기다리도록 하겠습니다. 혹시 뒤로 들어오시면 다 같이 박수 한번 이렇게 쳐주세요. 이거 또 가셨으면은 바로 이제 다음 뽑기 하면 되는데. 저희 설문조사 다 해주실 것 같고 혹시라도 카카오톡 저희 구독 이제 안 하신 분들이 계시면은 네 다시 뽑아야 된다고 하네요. 네, 1등 상품 저희 에어팟 프로 2세대입니다.
와이바이오로직스의 손종남 부장님, 네, 축하드립니다.
네, 그럼 저희 이제 당첨되신 분들이랑 기념사진 한 번만 찍을 예정이고요. 네, 딸기. Picture together. 감사합니다. 네, 이것으로 오늘 프로바이오 심포지움이 모두 마무리되었습니다. 끝까지 함께 해주신 모든 연자분들과 참석자분들 감사드리고요. 오늘 나눈 지식과 경험이 앞으로 연구와 협력에 큰 밑거름이 되기를 바라며 이후 여러분이 걸어가실 꽃길을 저희 프로바이오와 함께 걷는 길이 되었으면 좋겠습니다. 네, 감사합니다.